very specific at all because it's so broad. How can you be specific if it's a wide, broad group, right? So they're all telling us the same thing. So if I were to ask you something on fill in the blank, which, give me an example of a taxon that's the most specific or least specific, I hope these words don't throw you off, right? Okay? All right. So what's the answer here? What are my two least specific taxon in biology? Kingdom and domain. Very good. Or domain and kingdom if you want to go in order, right? Domain is more broad, right, than kingdom is. But technically speaking, they're the highest two, right? And over here, your lowest two or your most specific taxon, genus and species, right? You just go up here, you can go to, oh no, I did write it up here. Yeah, here and here, right? We won't talk so much about phylum, class, and order. I'll talk a little bit about families when I talk about virus families a little bit later today, but focus on the top two and the bottom two, which is what I'm going to talk about in a moment, right? Okay. So let's talk about this concept called binomial nomenclature. What is it and so forth? First of all, the prefix bi means two, right? So the two part is referring to a two-part naming system. Again, and I think I mentioned this before, for all living organisms, they all are identified by two names, right? That's what the bi, even us humans, we're animals, we have a two-part scientific name. I asked somebody one time, they're like, oh, is it human being? No, that's not, it's close, nice try, but that's not our scientific name. Okay, I'll tell you in a moment. And again, the scientific name for all living things is the genus name followed immediately by the species name. And genus is always capitalized. That's why I highlighted the G and capitalized. And then the species name is always lowercase, whatever, uppercase versus lowercase, what I'm trying to say, right? Capitalized, whatever, uppercase, and lowercase name. Okay. All right. So for these organisms, for example, and I did one already for you, right? It's down here. Let's go back to this leopard. I'm going to be also going back to this page. Again, the binomial nomenclature, the scientific name I told you for this animal is genus Pardus, genus Panthera, species Pardus, right? In that order. It's always genus first, followed by species name. Okay? And here's some other ones that I gave you. For examples, if we're talking about bacteria, because again, bacteria is a living thing, so it has to have, all bacteria have two-part naming system. So, and we've talked about this one already last time when we met, Escherichia coli, capital E, so it's my, what? Species or genus? Genus, lowercase c, species, right? You know this better as E. coli. Okay? When you write the official binomial nomenclature, the two-part naming system, it should be fully spelled out. Right? The genus should be fully spelled out. Okay? That's the preferred, correct way to write the binomial nomenclature, the name of all living things. You must spell out the genus name. However, in literature and journal articles, textbooks, they will introduce, like if it's an article about Escherichia coli, they will spell it out like first paragraph all the time, but then afterwards, it's actually permitted to start abbreviating. And the abbreviation is always, you abbreviate only the genus name, and then you must spell out the species. So it's always E. coli, but the real name really is Escherichia coli. Just like this guy here, we talked about this one before, last time when we met, about Helicobacter pylori. That's the official name to write it, but we also know this guy as what? H. pylori. So after a while, it's okay to start abbreviating later in the journal. They won't keep spelling the whole thing out. They assume you already understand that H stands for Helicobacter, just like E stands for Escherichia. Okay, but the official name, the correct way is really both names spelled out completely. Okay, and these are two bacteria. Okay. Nice little review. Which scientists work with H. pylori? We talked about that last time. We saw association with peptic ulcers and who? Barry Marshall, very good. Yeah. How about E. coli? If E. coli stays in the colon, it is called a what? A what? A resident microbe, okay. What's a resident microbe now?
It's a beneficial.
Okay, so what do we call that? What's the big term? term that we use? Is that a pathogen? No. So if it's not a pathogen and it's friendly and it's a good one, normal microbiota. That's normal. That's an example of normal microbiota. Okay, that stays in the colon. Okay, nice little review. Okay, another example here is this other organism called genus Penicillium, capital P, Chrysogenum, lowercase c. So you know it's genus followed by species name. And this is you probably already know this is a fungal mold, right? Because gee, I wonder what this mold produces. A drug called penicillin. We've talked about that last time. Which scientist accidentally discovered this because his bacteria plates were contaminated? It's okay, your exam's not till another 2 weeks, it's fine. You don't have to know first names, I'm probably not gonna help you. Alexander Fleming. Okay, Fleming didn't work with this, right? He discovered by accident. Remember, it had these plates were contaminated with a mold, and that substance they named it penicillin, right? And then therefore the mold they gave it the genus name Penicillium. Okay, very easy to remember, right? And here's our scientific name. We are called Homo sapiens. Okay, Homo sapiens. Later on, I'm going to talk about another species of genus Homo, but they went extinct a long, long time ago. Okay, we're the only ones left. I think we're the only animal with genus Homo. Okay, all right. So again, these are the full official two-part names of these specific type of organisms: a certain bacteria, fungal mold, and a human beings. Okay, any questions on binomial nomenclature? No? Okay. So I'm going to go back, like I told you, where's that delivered page? I'm going to go in detail. I'm going to start talking about the domains, and then after I talk about domains, I'm going to start talking about kingdoms. I'm going to skip everything in the middle, and then start talking about genus and species. That's how we're going to cover this. Okay, so let's talk about specifically more information about domain. Okay, so the domain system, this is just trivial. The domain system is fairly new. You probably think, well, that's like 30-something years ago. In terms of science, that's fairly new. Okay, prior to that, the highest kingdom used to be the highest taxa used to be just the kingdom. But now, since 1990, there's even a higher taxon that they now are calling the domain. Right? And all living organisms, all living things can be divided into 3 domains. And here are your 3 domains, right? Before I just told you domain, now I'm giving you names. There's 3 of them, right? So it's a 3-domain system. Domain archaea, domain bacteria, and domain eukarya. Okay? And you already know this domain is the highest of them all, right? The highest taxon. And again, this is for classification / identification purposes. Identification. Right, remember I told you when we classify things that the whole point is to identify them, right? Okay? And you already know that each domain consists of kingdoms, right? If you go back, sorry, I can go back to that record page. I should have just stuck it back in here. Dang it, I went too far. Where is it? Remember? There we go. Right, I told you several kingdoms make up a domain, right? That's why everything's indented to the right. Okay? So let's go back to slide 6. So yeah, each domain consists of kingdoms, right? So we're going to talk about kingdoms, not just yet, but in the next couple slides, we'll talk about some kingdoms here, all right? But each domain, consists of several kingdoms. And the domain name is really based, like these 3 here, they're based on the cell type of that living organism. And maybe you know this from basic biology, there are only 2 cell types, 2 categories, broad categories of cells. You're either a prokaryotic cell or prokaryote, or you are a eukaryote or eukaryotic cell. I don't know if you've heard of these terms before. You're probably more familiar with eukaryotes, right? From human anatomy and physiology, because humans, we're eukaryotes. I'll talk about that in a moment, okay? But these are the 2 cell types. There's no other choice. You're either a prokaryotic cell or a eukaryotic cell, okay? And the domain is based on which type of cell you are. And by that I mean, if you are archaea or domain bacteria, you're a certain type of cell. I think I have this on the next slide, right? So again, like I already told you, all cells, they're either prokaryotic cells or eukaryotic cells. And yeah, the prokaryotes, there are 2 of these 3 domains, and the eukaryote is one of them. And here's, I'll tell you the answer right here. Prokaryotes belong to either domain archaea or domain bacteria. These are prokaryotes or prokaryotic cells. Prokaryotic cells. And by default, if you're a eukaryote, then obviously you must belong to domain eukarya. So these are eukaryotic cells. Okay? The eukaryotic cells is very simple because there's only one domain you can possibly belong to, right? What's a little bit more problematic, if you're a prokaryotic cell, you kind of got a problem because you're one of these two domains, and as an organism, you only belong to one domain, you can't belong to two. So we're gonna have to differentiate between the prokaryotic cells to tell which domain they belong to, either archaea or domain bacteria. Okay? So again, this is kind of says a mouthful, right? It's like, oh my God, who's divided into what? And again, in my head, I see like a flow chart here, right? Again, rewriting the notes. So this is kind of pretty much what I just said on the other slide, right? Then I just tell you, you have two cell types. You have a prokaryotes and eukaryotes. And I told you in terms of domain, the domain eukarya consists only of eukaryotic cells, but these two domain, archaea and bacteria, they consist of prokaryotic cells, right? So again, this one is very simple. If my cell's eukaryote, then yes, I already know what domain that is. If it's a prokaryote, which domain is it? I don't know which one. I know it's one of these two, but yet I don't have enough information. I don't know yet. There has to be a way to distinguish which domain. Because again, this one prokaryotic cell cannot belong to two domains. It has to be one. So we're about to get into the differences between domain archaea and domain bacteria. We know the similarity. The similarity is what? They consist of prokaryotic cells. But there has to be a difference now between that population of prokaryotic cells in order to distinguish which one of these domains it belongs to. You can only belong to one of them. You can't belong to two. Okay? All right. And then I'll go back to this and add more information as we kind of go through some more slides. Okay? Let's talk a little bit more about the three-domain system. Okay? What's the origin of the prokaryotic cells and eukaryotic cells? Because I told you those are your main two cell types, prokaryotes and eukaryotes, right? And again, just to remind you that, and these are your domains here, right? Domain bacteria, domain archaea, domain eukarya. And remember these domains here, the bacteria and archaea, these are prokaryotic cells. And obviously domain eukarya, these are eukaryotic cells. Okay. But actually they had a common ancestor. That's what the origin is kind of telling us, right? And their origin, that ancestor that gave rise to all three of these domains, we're just calling that like LUCA. LUCA stands for the last universal common ancestor. So they did have a common ancestor up here. This is kind of like a tree, right? Like a little bit like a phylogeny, like we were talking about last time, right? Where the tree branches off and gives rise to different things, right? So you're gonna see here in a moment, like animals, I told you, animals are eukaryotic cells. So obviously they belong to domain eukarya, right? Okay, let me go back to that leopard page. Yes, indeed, domain eukarya, because animals always belong to domain eukarya. So kingdom Animalia belongs to domain archaea. I mean eukarya, right? So yeah, the leopard is an animal, right? Okay. Okay. So yeah, LUCA, the last universal common ancestor, that's the one that gave rise to everybody, right? To all the three different domains. Okay. Any questions so far? Good. Okay. So we're going to talk a little bit more about the three-domain system, and also we're going to talk about, I might as well introduce it here, I'm going to talk about the 5 kingdoms. Kingdom system. There are 5 of them, right? 3 domains, but 5 kingdoms. Okay? And we said already, again, I don't want to go back to the leopard, but we said domain, and then the subheading is a kingdom, right? Several kingdoms make up a domain, basically, right? And the kingdom that makes up the domain archaea and domain bacteria is this kingdom called kingdom Monera. Okay? Right, and then what cell type did we say domain archaea and domain bacteria consist of? Do they consist of prokaryotes or eukaryotes? Prokaryotic cells, right? Prokaryotes or prokaryotic cells, right? So are all members of kingdom Monera prokaryotes as well? They are because, yes, because if domain archaea and domain bacteria, they make up all members of kingdom Monera, then it's the same thing. All members of kingdom Monera are also prokaryotic cells. All right. So then let me go back to the slide 9. Let me go back to this one here. So if I had to now, because I gave you room here to write more stuff, right? So then if I had to fill this in terms of kingdom, I just told you these two domains here consist of members of kingdom Monera. And we said there's 5 kingdoms, so if I remember this is 1, and the order doesn't really matter, but there's 5 of them. These I haven't talked to you about. But there's 5 total, right? Only 1 kingdom consists of prokaryotic cells, which are these 2 domains. So again, I haven't talked about the differences yet about these 2 domains. Everything I'm talking about so far is what these 2 domains have in common. What they have in common are 2 things so far. They are all prokaryotic cells, and they all belong to kingdom Monera. Right? Again, we haven't talked about differences. We have to, we will. This is where this PowerPoint is going, right? And then the other 4 kingdoms that we have left, because there's 5 of them, right? 1 I already accounted for. I still have to talk about 4 more. Those kingdoms belong to domain eukarya. They're all eukaryotic cells, like kingdom animalia. Might as well put that one down here for you because we've already talked about that. Kingdom animalia. Okay, they're all eukaryotic cells. Okay, they're not prokaryote. They don't belong to these domains. Over here, animals do not. Animals belong to domain eukarya. Yeah, so, right, I just told you domain archaea and domain bacteria, they all contain members of kingdom Monera, and then I told you by default, the other domain eukarya, contains the other 4 kingdoms that we're about to talk about. One of these 4 is kingdom animalia, right? Okay. So I told you again, kind of sort of asked you this question in a different way a couple of times. I said here, if all members of kingdom Monera, right, this guy here, belong to domains archaea and bacteria, then how can you distinguish between the 2 domains? In other words, I'm asking you, what's the difference between domain archaea and domain bacteria? There has to be a difference. Otherwise, they wouldn't be 2 separate domains, right? There has to be a difference. That's what we're going to talk about next. Okay? So here I just kind of did a column to break things down for you, and we'll kind of go back and forth to see the differences between domain archaea and domain bacteria. Again, we know what they have in common. They are all prokaryotic cells, and they belong to kingdom Monera. That's all the similarities, all the things they have in common. We're about to talk about all the differences next, right? Let's talk about cell walls. The cell walls of organisms that belong to domain archaea and domain bacteria are very different, right? Let me talk about domain bacteria first, okay? Talk about the cell wall, okay? The cell walls of members that belong to domain bacteria have this substance that we call peptidoglycan. Okay, what is peptidoglycan? You'll hear more about it in the lab. But if you kind of break up this word, pept is referring to, does anyone want to guess? protein. It's referring to protein, and glycan is referring more to like the sugar. So it's kind of like it's like a protein with a sugar attached to it, right? Some compounds that's basically a protein with a sugar attached to it that is found where? In the cell wall of that organism. Okay? So, and I also should have given you a specific example here of domain, let me see here, 1 second. Yeah, a specific example here as well for domain archaea and domain bacteria. The example is bacteria. I know this is now starting to sound confusing because you have the organism bacteria, and then you have the domain bacteria, which are kind of two different kind of things. When I say bacteria, I'm talking about the actual organism. If I'm talking about domain bacteria, I will call it domain bacteria, right? But bacteria, these organisms in general, like what we work with in the lab and stuff like that, again, they are all what? Prokaryotic cells, and they all belong to kingdom what? Monera, all bacteria. Okay? And bacteria, you have to understand, we'll be talking about this in a couple weeks in the lab, bacteria have cell walls. Okay? Certain bacteria have peptidoglycan in their cell walls, so those bacteria belong to which of these two domains? Domain bacteria. But you can also have bacteria that belong also to domain archaea if their cell wall is different or unusual. And by unusual, I mean they are lacking peptidoglycan in the cell wall. And that's unusual. Most bacteria, almost all of them, should have peptidoglycan in the cell wall. Okay. The peptidoglycan gives structure and strength to the cell wall. We'll talk a lot more in lab about peptidoglycan in a couple of weeks. Okay, but for the most part, specific, the specific organisms that we know as bacteria, they have cell walls and they should have peptidoglycan in there. If they do, great, then we know they belong to domain bacteria, kingdom Monera. Okay? If they do not have peptidoglycan, they do not belong to domain bacteria. They are domain archaea. Kingdom Monera still, but domain archaea. So you see, that's one difference between domain archaea and domain bacteria, is the cell wall characteristics. Do you have peptidoglycan or do you not? Okay, that's one difference. You've already established a similarity. Similarities is they are prokaryotes and they belong to kingdom Monera. Let's look at habitats. Habitat is another way to differentiate between members of domain archaea or domain bacteria. If you belong to domain archaea, you kind of live in what they call extreme habitats, right? And they call these, for example, thermophiles. Thermophile, first of all, the suffix phile, does anyone know what phile means? Phile means loving, right? Therm is referring to what? Heat. So these are heat-loving organisms. In other words, they tolerate high temperatures, which is unusual, right? If something's really, really hot, you would think, oh, nothing's gonna grow there, you're gonna kill all the bacteria. Not if they are thermophiles. They actually love heat. Did you know you can still get bacteria, certain bacteria, obviously that belong to domain archaea, you can find them in hot springs, where the water's boiling. No, I don't know, Yellowstone, I've never been there, but whatever. I think there's hot springs there, right? At the foot of a volcano, people think it's too hot for a volcano, everything's gonna die there. Not if they're thermophiles. They love that. Actually, quite the opposite. If you remove them from the volcano and hot springs, you'll kill them. If you try to put them in a cooler environment, they will die. They won't tolerate that. They will only live in hot locations. That's unusual. That should destroy and kill bacteria. But I told you, they live in extreme habitats. So those bacteria, they belong now to domain archaea. And they're probably not going to have any peptidoglycan in their cell wall either. Okay? If you're a halophile, I told you, phile means loving. Halo is referring to salt. So these are salt-loving bacteria. Okay, salt-loving bacteria. tolerate and love salt. Again, most bacteria do not like salt. Okay? But this should not be anything new to you because we've talked about salt-loving bacteria before when we were talking about bioremediation. Do you remember that? What did they do to those? Where did they put the bacteria when we were talking about bioremediation in relation to halophiles? Well, those were halophiles. Where were they placed into? In the what? Toxic environment? Okay, there was toxins in there, but like, in terms of what type of water was that they introduced it to? Ocean. And the ocean is what? Salty. Right? Remember bioremediation? We took certain bacteria, placed them in the ocean to eat up or decompose the oil. Well, they thrive, they love that ocean. They can tolerate. Those are called halophiles. Most bacteria cannot tolerate salt. Okay, only certain minority of bacteria can, and those are the ones that are called halophiles, and those are the ones that can live in that environment. Okay. You also have halophiles can also live on the human skin. Our skin tends to be salty. Go ahead, lick your arm and see what does it taste like. No, I'm just kidding. Okay, because of sweat and all that other stuff, that's more physiology and stuff like that. But generally speaking, your skin is kind of salty a little bit. Okay, so we do have halophiles on our skin. They may be part of your like normal microbiota as well, because they probably don't cause harm to you. They actually protect you. Remember, they prevent the invading microbes from coming in, right? But again, this is unusual. Most bacteria are not halophiles. So all those that are halophiles, they belong to domain Archaea. Bacteria, there are some bacteria that we call methanogens, and these bacteria produce this gas called methane. Hence the name methanogen. But methane is a gas. Okay, and they produce and tolerate that. Most bacteria don't like methane gas. Okay, so then by default, how about domain bacteria in terms of their habitat? I just call it normal habitat because it pertains nothing like, if you're in domain bacteria, you cannot tolerate heat, you cannot tolerate salt, you cannot tolerate methane gas. Right? So those are some major differences. So now you see how we can, if we go back, where's my little flow chart? Well, again, here in blue, we already know what the similarities between these two domains are. Again, they're prokaryotic cells and they belong to kingdom Monera, but now we just finished talking for a few minutes now about the differences between them. I don't know if I have time to write them all up, but I mean, you can write them under here, right? You can have here, these were unusual cell wall. Didn't I just talk about that? Can I put CW for cell wall? Unusual cell wall, right? And then under, why were they so unusual? Because they had no peptidoglycan. I'll just put peptido, right? And over here was the opposite, right? Cell wall with peptido, right? Or yes, peptidoglycan, no peptidoglycan, right? And then in terms of habitat, we already talked about these were extreme habitat, and here was, I don't know what you want to call it, normal habitat. Dang it, sorry, pushed the button I should have. Where is it? Lost my paper. No, it's here. Sorry, I pushed the button on my pen. Oh, now it's doing this. Okay, never mind, sorry. Habitat, right? And the extreme one, I don't have room up here, but you know what I meant. The thermophiles, the halogens, not halogens, halophiles, sorry. Halophiles, thermophiles, and the methanogens, right? I don't have enough room, but you can put them up here, okay? So, you're rewriting things, which is good. It sticks in your head. I told you that one of the worst things you can do is just sit there and just go through the slides, PowerPoints, without writing anything down. Because you're working too hard when you're doing that. You're forcing yourself to memorize things. And, you have to ask yourself, if you just only do that, are you going to remember something from 3-4 weeks ago? Not unless it's better that you write things out on a separate paper, whatever, something mechanical, whatever. I'm just typing out or whatever. You guys have all these fancy pads that you're writing things out with. You can do that as well. Okay? All right. So again, are we clear with the difference between domain archaea and domain bacteria, right? Again, we know what they have in common, and the classic example is bacteria. So you have to look at that bacteria, okay, what type of environment does it live in? What does its cell wall look like? That's how I know which domain to place it in. If I don't have the information, I don't know which domain to place it in. The only thing I know is not domain eukarya for sure, right? That's the only thing I know. Okay, so let's talk about the kingdom now. We've talked about the domain. I'm not gonna go to the leopard page, but remember domain, and then we've talked about kingdom. I already told you there's 5 kingdoms total. The second highest, the highest one is the domain, right? What are they? We already talked about kingdom Monera, so we covered that one. So these other 4 belong, I told you, to domain eukarya. So the other 4 are kingdom protista, kingdom fungi, kingdom plantae, and kingdom animalia. This is slide 11, so I'll go back just one more time to the flow chart. Now you can fill them in, right? I'll just put K for kingdom, right? What did I say? Kingdom protista, kingdom fungi, kingdom plantae, and then I already wrote for you kingdom animalia. Okay, that's all domain eukarya, right? So let's talk about each of these each of these kingdoms. I already talked to you a little bit about kingdom Monera, and I'll still go over it again, but I'm gonna list like maybe about 2 or 3 bullet points for each kingdom, some highlights, and give you like an example of each. Okay, starting off with kingdom Monera. This is an FYI, I'm not gonna test you on it. Also known as, right, AKA means also known as, but this is just trivial. The trend in biology is now to, I don't know why, they make our life more complicated, to retire the word Monera. They want to replace it now with prokaryote. And they're gonna, I think, name it probably kingdom prokaryote. You know, I've seen it once, and at first I saw that, I'm like, oh my God, is that like the 6th kingdom now? No, they're just trying to retire, I don't know why. Kingdom Monera. When I was in school, I learned it as kingdom Monera. And now also they're saying no, that they want to name it instead kingdom bacteria. I'm thinking that's really confusing because you have bacteria, the actual organism, we have domain bacteria, and now they want to have a kingdom bacteria. It's too many bacteria. I would probably go for prokaryote instead because it starts confusing people. But in this class, I'm going to stick to Monera. You don't have to worry. Don't worry about these. I'm just FYI. If you ever see them, don't think it's a 6th kingdom. It's the same thing as Monera. Okay, so starting in order with kingdom Monera, right? You know there are 100% prokaryotic cells, right? And let me put also here semicolon. What domain or domains? Domain, is it just one? No, it was 2 of them. Domain archaea and domain bacteria. We just covered it. If you don't know it, that's okay, no big deal. No test tomorrow or anything like that, but eventually you do need to know things like that. Okay, so let me pause for a second. You know, week 2 of the semester, so far I think the material is very straightforward, isn't it? It's just a lot, and it's detailed. I told you, that's what nursing school and stuff like that, there's no secret to nursing school. It's not impossible, it's not hard. Or whatever, med school, PA school, it's not hard. I told you what's hard about it is keeping up and knowing it in a timely fashion, right? Because they don't mess around, they fly through the material real quick, and it's a lot, and it's detail oriented, just like here. So I'm hopefully getting you used to that. Okay, otherwise it's a piece of cake, it's not hard. The material itself is not hard at all. Okay, all right. They're unicellular, kingdom Monera, meaning they're very simple cells. It just consists of 1 cell, and that's it. There. That's a member of kingdom Monera, 1 cell. Or if maybe you want to make it this shape, sure, we can make it this shape. It's just 1 cell, unicellular, very simple organisms. And the example I already told you is the bacterium. Any bacterium, it has to automatically belong to kingdom Monera. Okay, so let me back up for a second because I want to stress this point because sometimes I use these terms interchangeably, right? We've talked about 2 cell types, prokaryotes and eukaryotes, and I told you prokaryotes on the previous slide, well, it's right here too. They belong to kingdom Monera. And the example is bacteria. So whenever I say prokaryotic cells, I am talking about bacteria. Because sometimes I won't say bacteria, I'll forget, I'm going to say prokaryotic cell. I'm assuming you understand a prokaryotic cell is a bacterium. I'm talking about kingdom Monera. Okay, so all of these three concepts, we're saying the same thing. Okay? All right. So yes, your example is a bacterium. We've talked about E. coli, Escherichia coli. Here's a picture of it, these rod-shaped cells. I guess the brown part is tissue or whatever, but you know, your specific example here in parentheses, these here are E. coli, Escherichia coli. You should know that. I will expect you to know that E. coli is a bacterium. It's not a fungus. It's not a virus, right? That's fair game, I think, because we've talked about this already a couple of times, like when we talked about normal microbiota. All right? That's next kingdom, kingdom Protista. So we said goodbye to prokaryotes, right? Now everything else, remember the other four kingdoms belong to domain eukarya. So these are all eukaryotic cells, and they're very simple eukaryotes. It's the simplest of all the four kingdoms. The other four kingdoms that we have left, right? Protista, fungi, plantae, and animalia. It's the simplest eukaryote. Simplest because most of them are unicellular. There are some that are multicellular, but for the most part, they just consist of one cell. And if you only consist of one cell, you're very simple. Okay, so very simple eukaryotes, so therefore mainly unicellular. And the example here is going to be the amoeba. Here's a picture of an amoeba. I think we're doing it today in lab, right? I see you in lab today, right? Yeah. So yeah, Kingdom Protista is our lab for today. So we'll be talking about amoebas today, but this is a specific picture. This is Amoeba proteus. Nice little review. What's the species name of this animal, of this organism? What is it? Which is Proteus, right? This is the species. I said species name? Yeah, species name, lowercase p, right? Genus name. Okay. Don't worry, I'm not going to ask you this on the exam. Okay, I'm not going to ask you, oh my God, was it Amoeba Proteus, Amoeba tigris? Who cares? Okay. What I do want you to know is amoeba is an example of a member of Kingdom Protista, though that is fair game. Okay. All right. And this is your amoeba here. So this is kingdom, our third, well, actually our fourth kingdom, because I already did Monera, right? No, I'm sorry. This what? Where am I at? Okay, Monera Protista. Sorry, this is the third one we're talking about. Yeah. Kingdom fungi is next, and again, they are eukaryotes. No surprise, right? I said I wasn't going to go back on some chillire. This is slide 13. Where's the flow chart? We're right here. I told you, they're all eukaryotic cells. They're domain eukarya. All of these, I'm going to have eukaryotic cell, eukaryotic cell, eukaryotic cell. I'm done with the prokaryote. Prokaryote was what? Kingdom Monera. So I'm done with that. Okay? Go back to slide 13. So yes. Now these are kind of half and half. Some are unicellular, some are multicellular. They're more complicated. Okay, half and half. The other ones that I talked about, like with Protista, the other one I told you was what? It was mostly unicellular. Now we see a transition where they start to become more multicellular, my eukaryotic cells. And members that belong to Kingdom Fungi, they can be unicellular, and some are multicellular. You can't be both at the same time. Someone asked a good question one time. No, either you're one or the other, but as a group, Kingdom Fungi, they're about 50-50. Okay, here are some broad examples and categories, and I think we've talked about this on day one, right? You can have the fungal mold, or you can have the yeast, the fungal yeast, right? Let's do the mold, because we've talked about this already so many times, right? Remember, that's where certain mold, this particular organism is the one that made penicillin, right? Accidentally discovered by Fleming, right? Okay. This particular picture, this is not a fungal mold. This actually picture is a yeast. Mold does not look like this. We have a mold lab. Actually, we have a Kingdom Fungi lab, not this week, but next Monday. We're doing Kingdom Fungi in the lab, okay? Yeast is another one, right? And here's an example, random example of a organism that belongs to the yeast category. It's a fungal yeast. called Saccharomyces cerevisiae, right? Now remember we had the, we were talking about, I think it was day one, we were talking about beneficial roles that microbes can do, like fungal yeast can do. Remember I told you that it had a lot to do with the alcoholic beverage industry. This particular yeast makes an alcoholic beverage. Anyone care to guess by looking at the species name in particular what it makes? Beer, because this is Latin based. Spanish is Latin based, so beer in Spanish is what? Cerveza. Cerevisiae. This is where it comes from. Latin based. So this particular yeast is what, this is how you get beer from this one. I'm not going to ask you where you got beer from. I'm just saying, let the words talk to you. Okay, it's not that hard. Whenever I see this, if I had no idea what it is, I'm thinking, beer, alcohol, oh, yeast. So it has to be a fungal mold. I don't even have to memorize it. I just have to look at words, right? I'm not going to ask you if this is mixed beer or whatever they're calling here. All right. I do care that you understand that yeast and mold obviously belong to kingdom fungi. All right. But in addition, I also want to tell you which one of these is multicellular and which one is unicellular. And I already told you about the temperatures. I think when I first introduced this, actually on our first lecture, right? So if I can do a little bit of a table here, and we'll do the yeast and the mold, do a table. And if we start talking about the number of cells, let's start with mold. Mold begins with M, and so does what else? Multicellular, right? So, gee, I wonder how many cells mold can take. They made our life so much easier. Thank you, scientists. Multicellular. Mold are multicellular, so by default, yeast are unicellular. Okay? Preferred temperature. Preferred temperature, I already went over that last time. Which one grows in cold? Mold, because it rhymes with mold, right? So this grows in cold temperatures, and this one prefers to grow more in warm / heat. Remember I told you even heat rhymes with yeast, kind of, sort of, maybe, right? Very easy to remember though. Guys, class so easy now, huh? Everything just makes sense, falls into place. Okay? Like I told you, this is yeast. This is unicellular. This is just one cell. Very different from mold. In lab next week, we're going to work more with mold. We don't really cover yeast so much. In the COVID lab, but we would obviously work more with yeast and stuff like that. Students, they took home little bags of baker's yeast and saw it under their microscope. Anyhow, all right. Okay, let's move on to the next kingdom, which is kingdom Plantae. Right? Again, no surprise, these are all eukaryotic cells. Domain who? Eukarya, right? And they are multicellular, very complex cells, because a plant is a complex organism, all right? And we also describe these as autotrophs. And what I mean by autotrophs are that these are self-feeders. They're called self-feeders. They provide their own food. For example, they will use the energy of the sun to make their own food. Remember photosynthesis, all that kind of good stuff, right? So I mean self-feeders, and I'll talk about later, versus the opposite, which is organisms that must consume other animals. How do you spell consume? Consume? Consume other animals. Plants do not eat other plants. Someone told me, how about the Venus flytrap? I don't know what they eat. I don't know. Are they even real? What do they really eat? I used to have one. I think it died on me though. But for the most part, plants are self-eaters. They make their own food. Okay, that's what you mean by auto. Doesn't auto mean self? Okay. Plants have cell walls, right? So geez, should not surprise that your example of a member of kingdom Plantae must be plants, right? Trees or whatever, right? Look at this big giant redwood. I guess they count the species name is appropriate, giganteum, because look at the people, right? And stuff like that. So, yeah, this is probably the giant redwood. Don't worry, I'm not going to ask you that. I don't care. That's not important. What's important is that plants belong to kingdom Plantae, very straightforward, and that they are autotrophs. They do not feed on other organisms. Unlike kingdom Animalia, which is going to be different and does feed on other organisms. No surprise here, they're eukaryotes, right? Domain eukarya. Also multicellular, right? Like plants, but even more complex than plants. So the most complex kingdom that we have. And again, they are this time heterotrophs. So they rely or whatever, they feed on other organisms for their food. They are not autotrophs, they are not self-feeders. Some random examples here. A worm is an animal, just like a fly is an animal. Here I'm just giving you some examples of a specific name of a roundworm is called Ascaris lumbricoides. You don't have to know this for this exam. I say for this one, but exam 3, when we talk about roundworm infections, you will. Okay, but don't worry about this one. And the housefly, the binomial nomenclature, the official name for the housefly is Musca domestica. Okay, I'm not going to ask you that. But again, Latin-based words. If you speak Spanish, what's the Spanish word for fly? Mosca. That's where the name Musca comes from the genus name. So you're learning not only micro, you're learning Spanish for free, so you didn't have to sign up for it. Okay, just to recap real quick, the differences between autotrophs and heterotrophs, again, has to do they produce their own food, their own energy source? Autotroph, yes. Heterotroph, no, because remember, heterotroph, it relies on another organism, right? And again, autotrophs, they produce their own food. And so, for example, via or through photosynthesis. Okay. And here, these are specific types of autotrophs or specific types of heterotrophs. We can call them photoautotrophs, chemoautotrophs. It just depends what they use as an energy source to make their food. I'm not going to go into detail with this because this is like another class. I'm going to let them teach you all about that. But don't write this down. You don't have to know. If you're a photoautotroph, you use energy from the sun to make your food. And if you're a chemoautotroph, you're using some other like carbon source from somewhere else to make your food. That's all it means. But just like heterotrophs, we also have photoheterotrophs and chemoheterotrophs. Basically, you have several types of these guys, is what I'm saying, right? And here are some examples. Plants you already know, because they probably undergo this process called photosynthesis. You probably already knew that. But also you're going to hear today in lab, also algae are capable of photosynthesis. And algae is not even kingdom Plantae. Algae you'll hear about today in lab, this is kingdom Protista. If you have a swimming pool, have you seen a swimming pool? A lot of times, there's different color algae, but you're probably coming with the green algae. You're probably thinking, and I hear this all the time, oh, algae, there's plant, that's plant. No, it's not. It's not kingdom Plantae, it's kingdom Protista you'll hear about today. You'll hear about lab why they're not plants. They used to be thought to be as plants, and they got kicked out of this kingdom Plantae and placed into their own kingdom. You'll find that out today. Okay? Examples of heterotrophs, organisms that rely on other organisms for their food, are things like herbivores, carnivores, omnivores. Herb is referring to plant. These are organisms that only eat plants. Carnivore, what do they eat? Carn in Latin, like in Spanish, carne is meat. So these are meat eating. I'm a carnivore, I love meat, so I'm a carnivore. I'm not a vegetarian, sorry, I can't do that. I'd starve to death if I was a vegetarian. Omni does both plants. Actually, no, I'm an omnivore, I do eat plants once in a while, but for me. Anyhow, omnivore is both, because there are some that will have both diet. Okay? All right. Any questions so far? Okay, pretty straightforward, right? So, this is slide 17. Where's my leopard? So we finished talking in detail about the three domains. We know how to distinguish between them. We know that they're made up of two types of cells, prokaryotes and eukaryotes. We've talked about the 5 kingdoms. Now I'm gonna jump, I told you I was gonna skip everything in the middle, and now I'm gonna talk about genus and species now. Right, so kind of finishing this theme here. So back to slide 17. Okay. So let's talk about the species, which is like the lowest of the low, right? The lowest taxon, right? And we know if it's the lowest taxon, is that least specific or most specific taxon? Most specific, right? Like tigress or pardus, you can't get more specific than that. If you're a pardus, you're a leopard. If you're a tigress, you're a tiger. Okay, you can't get more specific than the species. Lowest taxon, okay? And let's divide the species up in terms of our discussion. Am I talking about a prokaryotic species or a eukaryotic species? Okay, we're still gonna talk about species, but I'm gonna break them up and talk first about the eukaryotic species. The order doesn't matter. And then after I'm talking about eukaryotic species, I'm gonna switch over and start talking about prokaryotic species, but I'm still talking about species, okay? I remember I told you to get used to the term. Remember when I say prokaryote, I told you, aren't I talking about bacteria? Bacteria are prokaryotes, right? So when I get to eukaryotic species, I'm no longer talking about bacteria. It's something that I'm just calling non-bacterial. So for example, maybe an animal cell, because an animal is not a prokaryotic cell, it's a eukaryotic cell. Okay, so let me talk about eukaryotic species first. Okay, and again, these are not bacteria that I'm talking about. I'm talking more like animals, right? Okay, what's a eukaryotic species? Okay, species, but in terms specifically of a eukaryotic, right? These organisms, right, that are eukaryotes, you know, in terms of their species, they're closely related to each other genetically, okay? They're different, they're not the same organism, but they are closely related genetically to each other. And I'll give you an example, right? I told you human beings are known as Homo sapiens, that are still living like us, we're still alive, okay? Once upon a time, there was another organism called Homo erectus, who is extinct, no longer exists, okay? The Homo erectus, actually they were, you don't have to know this, they were here before Homo sapiens, and they were the first organism that looked kind of like humans that were able to stand up straight, because before they would hunch over, right? Or they would walk on their hands and feet, right? Kind of hunch, kind of like gorillas a little bit, sort of. Have you ever seen like a gorilla move, right? And we also have another one, I'll throw another one out. There's probably several of them. Homo neanderthalensis, Neanderthalensis. Have you heard of the Neanderthals? Cavemen. These are like the cave people. They call the Neanderthalensis, right? Okay. So you agree with me? These are three separate species, three separate organisms. If I have three separate species, these have to be three separate organisms. Okay, they're different, yet they are genetically related, closely related to each other. Because they have, you can tell by looking at the name, because all three organisms have the same what? Genus name. They all begin with Homo. They have the same genus names, so I know they're closely related to each other, but yet they are individually distinct because the three species names are different. It's like the same thing with Panthera tigris and Panthera pardus. I know these are two individual organisms, but they're similar. The tiger and the leopard are similar because they have the same Panthera genus name, right? And again, these are all three different organisms. There are some differences, but they have a lot in common, right? This is an artist rendition. I believe this is Homo neanderthalensis. You can tell very similar, but yet different. They had protruding forehead with like the eyebrows kind of out, the bone area, right? Eyes sunken in, and they kind of hunched over when they walked. It wasn't until we got to Homo erectus where they kind of stood up and didn't use their hands anymore to walk and things like that. And then obviously today, you know, we're different. But the other two guys, they went extinct a long time ago, right? And we're the only ones that have survived. Okay, but once some point of time, yeah, these were all here on Earth, they went extinct. Okay? All right. That was in terms of eukaryotes. Let's talk about prokaryotes next. So in terms of prokaryotes, again, no surprise, I'm talking about bacteria this time. That's my classic example. I'm not talking about animals anymore, like the cavemen and the whatever, Homo sapiens, and etc. Right? Here I'm talking about bacterial cells. So these are bacterial cells that have similar characteristics. For example, let's look at two species that belong to genus Streptococcus. Remember, the first name is the genus name, followed by the species name second, right? And I underlined the two species, right, for you here. So these are two species of the same genus, right? But these are both separate bacteria. Streptococcus pneumoniae, take a wild guess what that causes. Pneumonia, right? And there's another separate bacterium called Streptococcus pyogenes. You don't have to know this now. This is for example, we'll go into detail. Have you heard of strep throat? Pharyngitis? That's what this causes, strep throat, pharyngitis. We'll talk about that in exam 3, strep throat. And pneumonia, both of these, we'll cover these more in detail. But again, you agree with me that these are two separate organisms, right? Why are these, can someone tell me, why are these, by looking at the name, full name, how do you know these are two separate organisms? Because they have two different what? Species names. Are they closely related to each other? Yes, because they have the same genus name. But these are two separate species. Because they cause you can even tell me they cause two different diseases, right? Okay, because when we said pathogens cause disease, right? That was germ theory. I won't push anymore because we don't have time to be quizzing right now. I had to cover a lot of material. Let's look at this important term called clones next. Okay? Usually in microbiology, the term clone is only for prokaryotic cells. When we talk about clones, it doesn't really have, honestly, that much to do with eukaryotes, right? And a clone is basically like this population of cells that you get based on a single cell that divided. We call it the parent cell. And I'll show you what that means on the next slide with the hand-drawn diagram that I did. Okay? But clones are basically identical. They're like carbon copies. They're genetically identical. Basically, I'll just try it here. I guess I have room. You have the parent cell. Let's say I have a round bacterium. And this is my parent. We're calling it the parent because it's the originator. My parent cell. And this parent cell is going to divide into two. And it's genetically identical. So these are clones. I have two clones from my one original parent cell. They're genetically identical because they basically just split in half. Bacteria are unique. That's how they reproduce by splitting in half. Right? You don't have to know this now. That's asexual reproduction. It's unlike us. Is that how we make more of each other? Do we split in half? No, we actually have a whole different. That's sexual reproduction. Meiosis, mitosis, and all that stuff. Through mating and things like that, right? Bacteria do not mate. There's no male bacteria, female bacteria. Okay? Super important term. It's in bold, it's in red. Okay, very important to understand. All of you I know have heard of this word probably before in the past, strain, right? Another name for strain is the word subspecies. They're interchangeable. And sometimes they'll use this third word called serovar. They all mean the same thing. They're strain. Okay, they all mean strains. Okay. And a strain or whatever, a subspecies, whatever you want to call it, they start off as a clone, but after a while, something happened to the DNA. Remember, genetic mutation has to do with DNA changes. Remember I told you genetic comes from the word gene. Gene is a part of DNA, right? Something happens to that clone. I'll show you on the next page. But something happens to the clone where its DNA changes, and now the daughter cells, whatever the progeny of that cell, is not identical anymore, right? And that's how you end up with a subspecies / strain. Okay? And in microbiology or biology in general, you identify strains and subspecies by these numbers and letters that follow the species name, right? Remember, coli is the species, and now look, they added O157H7. When it has numbers and letters after the species name, that is a strain we're talking about. And by the way, strains and subspecies, they're bad. They're always pathogenic. They're pathogens. Remember, pathogens are capable of causing disease. So that is never a good thing. When you hear the word strain, that's not good. That's capable of causing a disease. There's no such thing as a good, friendly strain. If I would have just had E. coli by itself, then that could have been the normal microbiota. But if E. coli changes somehow, and now we call it O157H7, that's a strain, that's a pathogen. Okay? I give you another example here. Influenza A virus, the H1N5. I mean, who cares what these letters are, but as soon as I see letters and numbers, that indicates a strain / subspecies. Again, this causes the disease. Don't write this down, you have to know this. This is called the bird flu. H1N5 is responsible for the bird flu. That's why we don't have eggs, because of the bird flu. They had to kill all these chickens, hens or whatever you call them. Hens, I guess, right? Okay. This is kind of a mouthful, and it's a little confusing. You're like, oh my God, okay, I have to sit through and dissect it. That's okay, I did it for you. But yeah, technically you have to dissect it, because this kind of says a lot, right? And it's all written in words, and it can get confusing. At least for me, I don't learn this way. It's like, oh my, I get overwhelmed as soon as I see this, and I'm like, okay, I have to rewrite this. With pictures. Pictures are worth 1,000 words. Okay, so here's my artist rendition here of what I just said on the previous slide. So here we're talking about, sorry for this thing doesn't like glow in the dark, it's fluorescent. We're talking about prokaryotic species, cells. I'm talking about prokaryotic species, not eukaryotic species. So we start off with, like I told you, my parent cell. I'm just calling it E. coli because we know E. coli is a what? Bacterium. Okay? So here's my parent cell. E. coli divides in half and becomes 2 E. colis. Each one of those divides in half and becomes 2. So I went from 1, 2, 4. They're genetically identical, so don't we call these clones? I said on the previous slide, clone is genetically identical to the parent cell. Because basically they split in half and that's it. 100% same DNA. But sometimes now the DNA changes, right? Listen, I told you what genetic mutation means. DNA changes of the clone, and now instead of writing the blue clones of the blue circles, I purposely change them to red circles to indicate that the red is different from the blue circle. The DNA changed from the blue, and now it gave me this. These are no longer clones, these are now subspecies or strains. They are not genetically identical. Something happened that caused them to change. It's not important what happened right now, but something happened to cause them to change. And now these are no longer E. coli. Here in blue, these are still E. coli. Now this is the strain, so this is possibly E. coli O157H7. Again, we identify strains by letters and numbers that are associated after the species name.
So bacteria can be non-pathogenic if it's not strain, so once it becomes a strain, it's pathogenic.
Absolutely. She said, say it again. If it's the E. coli, if it's normal microbiota, is non-pathogenic for sure. And as soon as it becomes a strain, it is pathogenic. That's what you said, right? Yes. Very good. Very good.
Also, you had an example of the influenza A, and then the strain. So the influenza A was a normal.
Dang it.
Because it didn't sound like influenza.
Yeah.
I hear influenza A, that's a virus, right?
Yeah. Very astute of you to catch that. I'm gonna talk about viruses in a moment. Viruses, there's no such thing as a non-pathogenic virus. All viruses are evil.
Right.
The take-home probably that I should have said was, as long as you have letters and numbers, let's just say it's a strain.
Okay.
And strains are always bad for sure. The precursor to it may or may not be harmful. In this case, the E. coli one by itself is normal microbiota, but technically speaking, there's no such thing as non-pathogenic viruses. Good call.
That was just an example.
That was just an example.
Okay.
Very, very general example. probably should have said except for viruses.
Okay.
That's the exception. Okay, good catch. Speaking of viruses, let's talk about viruses a little, because viruses are really weird. There's this whole controversy that viruses are not even living things. And I'll tell you why in a moment, why viruses are considered non-living things. They're not cells. That's why they're not living things, right? And again, we already said that living things consist of cells, right? So, yeah, they're not cells, right? So, they use the term acellular, right? Because the letter A in front of a word usually means not, right? Acellular means not cellular. Just like, what does atypical mean to you? Not typical. What does asexual mean to you? Not sexual. A just means not. Okay, these guys are not cells. Why are they not cells? In order to be a cell, you need to meet two requirements. You must have a cell membrane, and they do not. Viruses don't have cell membrane, and they also do not have cytoplasm. You must have cytoplasm in order to be a cell. They don't have that. Okay, couple weeks we have the virus chapter. That's our last chapter in this exam. And we'll study the structure, and I will never ever mention cytoplasm or cell membrane. They're just completely made up of something completely different. They're weird. No one even knows where they came from. Okay, but yeah, they're not cells because they don't meet these requirements. So are cells prokaryotes? I'm sorry, cells. Are viruses prokaryotes? What's a prokaryote? Is it a type of cell? Yeah, so I just told you they're not cells. So a virus cannot be a prokaryote. Can it be a eukaryote? No, because eukaryote are also types of cells. These two are my two types of cells, remember? We just went over that. So a virus is not a cell, so therefore a virus cannot be a prokaryote or eukaryote. So do you understand why the controversy people say, then they're not living things? And it's true, they really aren't, because living things consist, as the first thing I said, living things consist of either a prokaryotic cells or eukaryotic cells. You must meet that requirement to be a living thing. They're not cells. They're not living things. They're weird. Like, so weird. Okay, but they're not living things. True or false? Viruses can be categorized into a domain. Can a virus be categorized into a domain? What are domains made out of? Kingdoms. And kingdoms consist of cells? Do kingdoms consist of cells? Are there cell types that make up a kingdom? And all that stuff, the domains? Are there cells that make up the domain? Like, remember I told you domain, archaea and bacteria are prokaryotic cells, and domain eukarya was eukaryotic cells. So viruses cannot have a domain either, because to be a domain, you're one of these two type of cells, and you don't have a kingdom either, because if you don't have a domain, can you have a kingdom? No, they're not cells. Okay, so they're just really weird. So no domain, no kingdom, because they're not cells. Why aren't they cells? Because they're not prokaryotes and eukaryotes. Why not? Because they don't have cytoplasm and they don't have cell membrane. That's why. Just really weird organisms. They're even more weird because they're described as obligate intracellular parasites. Obligate, the word obligate is kind of to me means like an obligation, like you must do something, right? So they must invade. Intracellular means you go inside the host. So you must invade a host cell, like the human host cell. Why do they invade the host cell? In order to multiply. In order to multiply. Viruses are so lame, they can't even multiply on their own.
So they have to feed off of
They have to feed off, invade a host cell in order to multiply. That's why they are obligate, intracellular, inside the host cell. What's a parasite? Parasite takes advantage of a host cell and destroys it. And viruses are super evil. That's what they do. They will invade that host cell, and guess what happens to that host cell? You think that host cell's gonna live? No, it destroys the whole cell. They're absolutely evil, right? So when I get to the next chapter, not next chapter, the last chapter for this exam, I'm gonna give you the example. It's like the home invasion. I probably shouldn't be talking about things like this right now, but it's like a home invasion. They come to your house, take you hostage, abuse you, this and that, and then it's a group of them, and they mate with each other, and they make more babies of themselves. And when they can't fit in your house, they leave and then blow up the house with you in it. And they move to the next house and do the same thing and make more of each other, multiply, mate, and make more and more. That's what viruses do. They will destroy the whole cell at the end, once they're done using it. Isn't that what a parasite is? It's a one-way relationship. Parasite takes and takes, and at the end damages the host cell. And the host gets nothing out of it. Quite the opposite, the host dies at the end. Okay, that's why viruses are evil. We'll talk about viral diseases and all that, but we have to understand what a virus does. And this is just general. I'm going to get more into detail. There's a lot more in detail about even how it invades a cell. A lot more detail that we're going to get into in about 2 weeks. Okay? How do you classify viruses? Viruses can be classified, and their highest domain is the family. That's where they start from. There is no domain, no kingdom, but there is a family. And then I'm not going to go back to the leopard page, but if you see it's family, genus, and species. Okay? And the family name of viruses, they all end with the letters that spell out viridae. So retroviridae is the retrovirus family. Herpes viridae is herpes virus family. All the families end in viridae. Okay? And you can also have, like we've just talked about a little bit earlier, like what you mentioned about the influenza, H1N1, all that stuff. You can have strains of viruses as well. Okay, and again, if they follow, if the name is followed by letters and numbers, then you know that these are strains, right? So we can have the influenza, influenza A, H1N1, right? And then whatever, the H1N5, all these things, right? And there's probably a lot more, right? H1N1, by the way, FYI, you don't have to know, I'm not gonna ask you, is the swine flu. Swine is a pig flu. What's an avian? Bird, right? This is the bird flu, the H1N5. Okay. Again, you don't have to memorize these numbers. And was it N2 and H17? Who cares? The bottom line is that if you have a mixture of letters and numbers, that's a subspecies or strain. And viruses have them as well. Okay? Yeah, depending. We'll get in exam 3 we will talk about this in more detail in exam 3. The numbers will matter that because you'll have to distinguish what type of flu it is. Is it a pig flu or bird flu? There's several types of other types of fluids as well. Okay. One thing that I just mentioned, let me go back for 1 second. When I first introduced this topic about species and subspecies, right? So we're used to looking at the bottom 2 taxa, right? Genus followed by species, and I told you species was the lowest taxon. I'm glad I remembered, right? Subspecies is not a taxon. So don't get that confused, right? Because if you kind of start writing them out, it's genus name, and then species name. Species, again, I'm telling you, it's still the lowest taxon. Technically speaking, the subheading of species obviously is subspecies, but subspecies is not considered a taxon. That's one thing I almost forgot to mention. Okay, species is still the lowest taxon. This is just a different classification, but it's not considered actually a taxon. Because this is due to, the subspecies got created, remember, because of genetic changes. So the species still remains the lowest taxon. I'm just telling you that because I sometimes have people that start telling me that subspecies is the lowest taxon. It's not. It's not even a taxon, period. Okay, I almost forgot about that one. Okay, so let's talk about next methods of classification. Like ways that you can classify. We can distinguish when something has been classified. We already talked about how to classify, how to get a 2-part name right? But here's some ways in which you can do that, right? How you can tell, it helps you identify what type of microbe you're dealing with. You can look at structural or morphological changes or characteristics, right? And by that I mean looking at the shape of that organism. Like bacteria, for example, are they round or are they rod or are they spiral shaped, right? The arrangements, are they arranged by themselves, like single, or are they arranged in clusters? Are they arranged in chains? We'll talk a lot about this in lab, about arrangement, shape, and stuff like that. And today in lab, we're going to talk about size, how to measure things with the ocular micrometer, right? So all those things gives you information so that you're able to classify and identify, okay, based on morphological differences. Remember, you have to, in the microscope, you have to be able to see the bacteria. I mean, you can't tell the shape if that bacteria is not stained, right? If it's colorless, you won't be able to see it. So staining is very important, right? And there's a staining procedure that we call gram staining. You'll do this a lot in lab. And with gram staining, you're only staining the cell wall. Gets stained. The cell wall gets stained. The cell wall of the bacteria. And depending what color that cell wall ends up being, that helps you identify, distinguish which bacteria you're looking at. Right? All right. And when we're doing gram staining, our end result is either going to be recorded as a gram-positive bacteria or gram-negative bacteria. The gram-positive bacteria, you end up with purple cell walls. Can I put CW for cell wall, you remember? These are gram-positive are purple cell walls, gram-negative are red/pink cell walls. Okay, so when you do gram staining, those are your two colors that you're gonna end up looking at, right? Or we just call them gram-positive bacteria or whatever, you know, but it's basically the bacterial cell wall is picking up a purple stain. Over here, the negative bacteria is picking up the red/pink dye that we use. So this is all staining procedures. And you'll get to do this in lab. In fact, your demonstration quiz is based on gram staining that you do like week 6 or whatever that is. Okay. And again, what's the difference between a gram-positive /purple cell wall and a gram-negative /reddish-pink cell wall? It's the amount of peptidoglycan. I didn't mean to scratch that out. The amount of peptidoglycan in the cell wall. Remember we talked about cell wall a couple minutes ago earlier today? That there's peptidoglycan in the cell wall for most bacteria, right? Only if you belong to domain eukarya. We said they have that. Remember the unusual cell wall, that was the other first two domains, domain archaea and bacteria. Okay, we talked about all this today, right? I don't expect you to know it all because I just covered it, right? You still need to cover it on your own, right? Okay. You can also run biochemical tests to help you distinguish between different types of microbes or organisms, right, for identification purposes. And what you can do is you kind of create this. I mean, you can do these. Yeah, you run these biochemical tests, and you create something. I don't know what you want to call this. Like what's the right word I'm looking for? Like some sort of tree, right? So in other words, for example, let's say, does that bacterium use glucose or not, right? And there's two answers, right? Either yes, it uses glucose, or no, right? And then we can run, so let's say my bacteria uses glucose, and I go this route, so I'm using this part here, and then the next question would be, does it use lactose? I don't know, another sugar, right? And then you go again, yes or no. If it does use lactose as well, so it uses glucose and lactose, then maybe under yes, this can be E. coli. So my final answer would be E. coli, based on these chemical tests. If it was no, maybe it's H. pylori. I don't know, I'm just making stuff up. I think I misspelled pylori, you know what I mean, H. pylori, right? So biochemical tests are very, very useful. That's what your unknown project I talked to you about, that's what it all is. You're doing all these biochemical tests and narrowing it down and eliminating what it cannot be. So this is very helpful. These paired questions of yes or no questions. Do you, does it grow in oxygen? Whatever, yes, no. If it's no, then we only stick to the possible candidates it can be. If it's the yes, then we eliminate everything else because we're not dealing with that. Yeah, so this is why I tell you that. This is the example, right? Can the unknown break down glucose? Yes or no, etc. Okay? All right, so I just want to finish this PowerPoint plus I had you for a lap, so you're not in a hurry to get home because you're not going home right now anyway, right? So it's okay, I don't think there's a class in here anyway. So it's just a couple more slides, like two or whatever. Okay, serology is the next type of method that we can use. Serology comes from the word serum. Have you heard of serum? Well, it's right here, blood serum. It's that clear part of the blood. If you spin the test tube full of blood, like what they do when you go for blood work, phlebotomy or whatever, it's that clear part of the blood. Okay, so we can do blood work to help us identify. Blood tests to help you identify what type of bacteria, for example, you have, right? And it's really good for differentiating like what type of strain you may have, okay? Because they look at the DNA and so forth, and they're able to tell maybe what strain that you have, okay? And that's based on blood work. Okay, they're looking for that pathogen. Once they find it, they get the DNA from it and analyze the DNA and tell you your results based on their catalog of what that DNA is. Okay? So here, when they look at the DNA sequence, again, they can distinguish, for example, between the non-pathogenic E. coli and the pathogenic one, right? The non-pathogenic E. coli is just, regular E. coli, which we were calling normal what? Non-pathogenic normal microbiota, right? The pathogenic one, let's stick to E. coli. Am I just gonna write it like this and that's it, if it's pathogenic? No, I have to add letters and numbers to it, such as O157H7. Okay. This one here, again, this is pathogenic. This one will cause illness and disease. This one will not, as long as it stays in the colon. Okay. And that can tell us through blood work by looking at the DNA of that unknown organism which one of these it is. Is it pathogenic or non-pathogenic? Okay. Number 5 out of 6. So a next method is called PCR, which stands for polymerase chain reaction. And what PCR basically is, it basically makes or amplifies many copies of DNA. It just mass produces DNA. Mass production of DNA. Sometimes you need a lot of DNA because you need to run a lot of tests, for example, and this and that, right? But it's like a copy machine for DNA to make lots of copies of it, right? You start off with a single one, and you make a lot of copies. Here, I'm not going to get too much to it, but this is the best picture I can find. But this is your original DNA, right? DNA, if you didn't know, is usually double-stranded. It's connected here in the middle. Looks like a ladder. They untwisted it. Usually it's twisted like a ladder. It's twisted, right? And they started off with one DNA, and look, over here they ended up with 4. They mass-produced it. Don't worry about this other stuff. It's not important. It's the wrong class for that. You can take molecular or whatever, genetics, if you want to know all the details of that. Okay, but PCR is also very useful to make lots of DNA. The more DNA, the more tests you can run, the more answers and results you can get from this. Okay, our last method is using textbooks or manuals, and here's an important textbook. It's called Berge's Manual of Determinative Bacteriology. Okay. You will get very, very familiar towards the end of the course with this manual because this is what you use. This is the reference book that you use for the unknown project. Okay. Remember the unknown that I give out like week 11, you'll be using this for copies of it, photocopies. Right? And basically this Berge's Manual is basically the Bible of bacteria. Everything you wanted to know about bacteria is in that book. It's all about bacteria only. And it helps you to identify unknown bacteria. Okay? We're still behind schedule, I know, but we will catch up. I think we should have been on chapter